Biochemical test for gram-negative bacilli. In this video, we are going to discuss the principles behind the biochemical test used for the identification of gram-negative bacilli.
First, we have the triple sugar iron agar test, or TSI. The TSI agar test is used for the identification of enteric bacteria. These are bacteria belonging to the family of Enterobacteriaceae. It is also used to distinguish Enterobacteriaceae from other gram-negative intestinal bacilli by their ability to catabolize glucose, lactose, or sucrose, and to liberate sulfides from ferrous ammonium sulfate or sodium thiosulfate. TSI agar slants contain a 1% concentration of lactose and sucrose and a 0.1% glucose concentration. The pH indicator, phenyl red, is also incorporated into the medium to detect acid production from carbohydrate fermentation. So here are the tubes of TSI tubes with different reactions. So how do we inoculate the TSI tube? So if this is your TSI agar tube, then you stub the butt using your inoculating needle, and for the slant, streak it using a zigzag pattern, and then incubate for 18 to 24 hours in order to detect the presence of sugar fermentation, gas production, and hydrogen sulfide production. There are different reactions that may occur in the TSI tube. So we have a yellow bot and a red slant. It is written as K over A. The numerator refers to the slant and the denominator refers to the bot. So this reaction is due to the fermentation of glucose, in which phenyl red indicator turns yellow due to the persistence of acid formation in the bot. This slant remains red or alkaline because of the limited glucose. Remember, we only have 0.1% of glucose compared to the other sugars present in the medium, and therefore limited acid formation which does not persist. So this is another reaction that may occur in the TSI tube, a yellow bot and slant. So both are yellow, both are acid, so it is written as A over A. This is due to the fermentation of lactose and or sucrose. So the yellow slant and bot is due to the high concentration of these sugars that lead to excessive formation of acid in the entire medium. Gas formation is also detected in the TSI tube, which is noted by the splitting of the agar, or this may be noted by a crack or a space at the bottom of your tube. So if your agar is raised, so this space indicates that there is a gas formation in your TSI tube. So H2S or hydrogen sulfide gas formation is also detected by the formation of black stuff, or if there is a blackening in the agar, it means that the organism is H2S positive. For the last reaction, we have a red bot and slant. Both are red and it is written as K over K. It indicates that none of the sugars were fermented and neither gas nor H2S were produced. So how do we report and interpret the results? If there is a black bot, so it means that it is H2S positive. No block in bot, H2S negative. If, if gas is produced, so the letter G is written in your result. And if there is a yellow agar, it means it is acidic. So it is written as A. And for red agar, it means alkaline. It is written as K. The next set of tests is called the IMViC test. This is a mnemonic to remember the four biochemical tests being used. Those are indole, methyl red, Vogues Proskauer, and citrate. These four tests help divide the Enterobacteriaceae into two major groups, the E. coli group and the Enterobacter Klebsiella group. So let's talk about the indole production. Almost all proteins contain the amino acid tryptophan. Bacteria containing the enzyme tryptophanase can hydrolyze tryptophan into indole, pyruvic acid, and ammonia. Pyruvic acid and ammonia are being utilized by bacteria as nutrition, while indole is not utilized and therefore accumulates in the medium. The presence of indole can be detected by the addition of Kovacs reagent. Kovacs or Ehrlich's reagent reacts with indole producing a bright red compound on the surface of the medium. Bacteria producing a red layer following addition of Kovacs reagent are indole positive, while the absence of red color indicates tryptophan was not hydrolyzed and the bacteria are indole negative. So the SIM or SIM or sulfide indole motility tube medium is the one being used to detect the indole production. Next, we have the methyl red test. So for this test, we have methyl red as our indicator. At low pH, it turns red or it remains red and turns yellow at high pH. All enteric bacteria catabolize glucose for their energy needs. So this glucose is converted to pyruvic acid via the Embden-Meyerhof pathway. And this will further be converted to other compounds depending on the enzyme pathways present in the bacteria. Some bacteria, like the Escherichia coli, which is a mixed acid fermenter, enters the mixed acid pathway and converts this pyruvic acid to stable acids like lactic, acetic, and formic acids. So mixed acid fermenters such as E. coli produce a mixture of fermentation acids and thus acidify the medium. So let's say this is your MR tube. So if your bacteria entered the mixed acid pathway, stable acids will be present in your medium and thus after the addition of methyl red, it will turn red due to the acidic environment, indicating a positive test. But if the organism inoculated in your medium wasn't able to convert this pyruvic acid into stable acids, your medium will turn yellow upon the addition of methyl red, and it indicates a negative test. This test is of value in distinguishing Escherichia coli, which is a mixed acid fermenter, and Enterobacter aerogenes. So this is an actual demonstration of MR test. Again, presence of yellow color indicates a negative test, while the presence of red color indicates a positive test. For the Vogues Proskauer test, this test identifies bacteria that ferment glucose to 2,3-butanediol accumulation in the medium. The same with methyl red test. First, we start with glucose that would be converted to pyruvic acid via the Embden-Meyerhof pathway. But instead of entering the mixed acid pathway, some bacteria enters the butylene glycol pathway, converting pyruvic acid into acetoin. Now, for example, this is your Vogues Proskauer medium inoculated with your organism using your wire loop. So you add your 40% potassium hydroxide reagent. So what will happen? Acetylene will be converted to diacetyl following the addition of potassium hydroxide. And this diacetyl will be detected using a 5% solution of alpha naphthol in absolute ethanol, or it's also called Barrett's reagent. It will detect the presence of acetylene or acetylcarbinol, which is a precursor in the synthesis of 2,3-butanediol. So a red color is indicative of acetylene production, representing a VP positive test. Absence of red color is a VP negative test. Now we have the citrate utilization test, the last test for this IMViC test. The citrate utilization test is used to determine the ability of the organism to utilize sodium citrate as its only carbon source and inorganic ammonium salts as its only nitrogen source. For this medium, we have bromthymol blue as our pH indicator. So it is green at low pH and turns blue at high pH. So we have sodium citrate incorporated in the medium. Now, if our bacteria has this enzyme citrate permease, the citrate will be utilized and be converted to pyruvic acid, oxaloacetic acid, and carbon dioxide. Since citrate has been utilized, sodium will be liberated and will now combine with the newly formed carbon dioxide and water. So it will form sodium carbonate, which is an alkaline compound. So since this is an alkaline compound, the bromthymol blue indicator will turn from green to blue, which indicate a positive test. So this is an actual photo of a citrate tube. The green one is a negative test, while the blue one indicates a positive test. So that's all for the IMViC test. Now we have the urease test. For the urease test, we have phenol red as our indicator. So it is yellow at acidic pH and turns pink or bright red or purple at high pH. Some bacteria are able to produce an enzyme called urease that attacks the nitrogen and the carbon bond in amide compounds such as urea, forming an end product ammonia, carbon dioxide, and water. So this medium contains urea, and some bacteria have this enzyme urease, in which urea is converted to carbon dioxide, hydrogen, and ammonia. So since ammonia is an alkaline compound, it will turn your medium to pink or let's say red or purple, deep pink or purplish red, and it indicates a positive test. So the urease test is used to distinguish members of Proteus from other non-lactose fermenting enteric bacteria like Salmonella and Shigella. Urease activity is detected by growing bacteria in a medium containing urea and using a pH indicator such as phenol red. So when urea is hydrolyzed, ammonia accumulates in the medium and makes it alkaline. And this pH increases, or this increase in pH causes the indicator to change from orange red to deep pink or purplish red, and it is a positive test for urea hydrolysis test. Failure of a deep pink color to develop is a negative test. So are actual tubes for urea test. We also have phenylalanine deaminase test. This test can be used to differentiate among certain enteric bacteria such as E. coli and Proteus vulgaris. Proteus vulgaris produces the enzyme phenylalanine deaminase, which deaminates phenylalanine to phenylpyruvic acid and ammonia. When ferric chloride is added to the medium, it reacts with phenylpyruvic acid, forming a green compound indicating a positive test. So this is, let's say this is your medium for phenylalanine deaminase test. So you inoculate your slant with your organism and then add your ferric chloride to detect the presence of phenylpyruvic acid, which indicates that the organism has the ability to deaminase phenylalanine. So if it is positive, it will form a green compound. So since E. coli does not produce the enzyme, it cannot deaminate phenylalanine, hence there is no change in color, so indicating a negative test. Lactose fermentation test. For this test, we have a lactose broth with Durham tube, which is a smaller tube inverted inside a larger tube within the culture medium. So organism will be inoculated using your wire loop, and phenol red is also used as our pH indicator. If a fermenting bacteria are grown in a liquid culture medium containing lactose, they may produce organic acid as byproducts of the fermentation. These acids are released in the medium and lower its pH, turning your phenol red to yellow because of the acidic environment. Gas produced during the fermentation process can be detected using a small inverted tube, so the Durham tube within the culture medium. And if gas is produced, the medium inside the Durham tube will be displaced and trapping the gas in the form of a bubble. So you see that space in the Durham tube indicates a gas production. Lysine decarboxylase test is used to determine whether gram-negative raw decarboxylates or deaminates lysine and forms hydrogen sulfide. So lysine iron agar test. Agar is the medium being used for the detection of decarboxylation of lysine. It contains lysine, peptones, a small amount of glucose, ferric ammonium citrate, and sodium thiosulfate. So this is inoculated by stubbing the butt and streaking the slant in a zigzag pattern. When glucose is fermented, the butt of the medium becomes acidic or yellow due to the low pH. If the organism produces lysine decarboxylase, cadaverine is formed. Cadaverine is a medium or it's a compound that neutralizes the organic acids formed by glucose fermentation, and the butt of the medium reverts to the alkaline state or purple due to the increase in pH. But if the decarboxylase is not produced, the butt remains acidic or yellow. Now, if you see your lysine iron agar tube with yellow butt after overnight incubation, it indicates that the organism is negative for lysine decarboxylase. For the determination of the amination of lysine, so we have if oxidative deamination of lysine occurs, a compound is formed that in the presence of ferric ammonium citrate and a coenzyme flavin mononucleotide forms a burgundy color on the slant. So first, glucose will be fermented, turning your butt to yellow, and if it's positive for deamination, your slant will turn to burgundy color, indicating that it is positive for deamination. For the sulfide indole motility test, or SIM test, we have this tube and it is inoculated using your inoculating needle stub at the center. And then when proteins that are rich in sulfur containing amino acids such as cysteine are hydrolyzed by certain bacteria, hydrogen sulfide is produced. The SIM medium contains peptones and sodium thiosulfate as substrates and ferrous ammonium sulfate as H2S indicator. Cysteine is a component of the peptones used in SIM. Once H2S is produced, it combines with ferrous ammonium sulfate, forming an insoluble black ferrous sulfide precipitate that can be seen along the line of the stub inoculation. If the organism is also motile, the entire tube turns black. The black line or tube indicates a positive H2S reaction. Absence of a black precipitate indicates a negative test. SIM agar may also be used to detect the presence or absence of motility in bacteria as well as indole production. Motility is present when the growth of the culture is not confined to the stub line of the inoculation. So you see this cloudy part surrounding the line of inoculation. It indicates that your organism is motile. Growth of non-motile bacteria is confined to the line of inoculation. To test for indole production by the bacteria, Kovacs reagent is added to the SIM cultures and observed for the development of a red color. Oxidase test. This is a useful procedure because some gram-negative pathogenic species of bacteria, such as Neisseria gonorrhoeae, Pseudomonas aeruginosa, and Vibrio species, are oxidase positive, in contrast to the family of Enterobacteriaceae, which are oxidase negative. Oxidase-positive bacteria have enzyme cytochrome oxidase, which is able to oxidize the substrate tetramethyl paraphenyline diamine dihydrochloride, forming a dark purple product indophenol. The ability of bacteria to produce cytochrome oxidase can be determined by the addition of the oxidase test reagent to colonies that have grown on a plate medium. Or you may also use a wooden applicator stick. A bacterial sample can either be rubbed on a filter paper moistened with the oxidase reagent. The light pink oxidase test serves as an artificial substrate, donating electrons to cytochrome oxidase and in the process becoming oxidized to a purple, then dark purple compound in the presence of free oxygen and the enzyme oxidase. The presence of this dark coloration represents a positive test. No color change or a light pink coloration on the colonies indicates the absence of oxidase and is a negative test. Positive test. So here are examples of biochemical results for Enterobacter aerogenes. So can you help me interpret the biochemical results? So look at the TSI tube. The first tube, we have a yellow slant and yellow bot, and there is a space at the bottom indicating a gas formation. So it will be written as A over AG because there is a gas formation. Next, for the LIA, we have two, the carboxylase and the aminase. So if it is positive for the carboxylase test, the bot should be purple. So it's purple, that's why it is positive. And if it is positive for the aminase, the slant should be burgundy. But it is not, so it is negative. Citrate tube is blue, so therefore it is positive. For the seam, so it is not clear, but it is negative for sulfide because there is no black formation in our seam medium. For the indole, there is no pinkish compound at the top of our medium, therefore it is negative. Motility, so I don't know if you can visualize it or if you can see it, but it is motile. For the MR, so this is yellow, it means negative. VP is also yellow, it means negative. The urease is also negative. Next, we have the E. coli. So E. coli, again, A over A, Lea positive, decarboxylase positive, deaminase negative, citrate negative, sulfide negative. So you can see at the seam medium, we have the formation of pink color above the seam medium indicating a positive indole reaction. Motility is negative. MR, so if you compare the previous example, we have red here, so this is MR positive. VP negative. Urease negative. Next we have Klebsiella. So Klebsiella is A over AG. Lea is positive. Deaminase negative, citrate positive, sulfide negative, indole negative, motility negative, MR positive, VP positive. So you can see that the reddening in the upper portion of the VP tube, and urease positive. So we have pink. Approaches vulgaris, we have so red slant, so K over A plus H2S. And for the Lea, so it is yellow. The butt is yellow, therefore it is negative for the carboxylase, but the slant is burgundy, so it is positive for the aminase. Citrate is positive, sulfide is positive, indole positive, motility positive, MR positive, VP negative, and urease positive. Last example, we have the salmonella. For the TSI, we have K over A, and there is a blackening here, so it indicates H2S production. So it will be written as K over A plus H2S. The carboxylase positive, deaminase negative, citrate negative. So for the seam, the sulfide is positive, indole negative, motility positive. MR is positive, VP is negative, and urease negative. So I hope you enjoyed this exercise and you have learned the principles of the biochemical test used for the identification of gram-negative microorganisms. Thank you.